Hello, everyone, and welcome to the third seminar of BioElevate. Today, we are very excited to have Nicola Ria, a PhD student from Catalan Institute of Nanoscience and Nanotechnology. He is a PhD candidate working on a new biomedical nanotechnology for the treatment of movement disorders. His work is divided into two main benchtop deep brain stimulation for Parkinson's diseases and electrical static nerve stimulation for spinal cord injuries. So today, Nicola is going to give a talk on graphene-based flexible neurotechnology for high-resolution bidirectional neural interfaces. So now, before I go to Nicola, just a quick note that if you have any questions, please write them in the Q&A box or raise your hand at the end of the talk. You can open your camera and microphone as you like, but we are going to take all of the questions at the end of the talk. So now, without any further delay, so let's go to the Nicola. And Nicola, please, the floor is yours.
So, good afternoon, everyone, and thank for the kind introduction. Today, I'm going to talk about my work about graphene-based neurotechnology for high-resolution bidirectional neural interfaces. So, neural interfaces are devices which interface directly with the nervous system in order to improve the fundamental understanding of normal and diseased brain state. To find new biomarkers and modulate them in the case of Parkinson or epilepsy by placing cortical electrodes on the surface of the brain or deep brain implant. Or neural interfaces are also neuroprosthetics used for motor and sensory disabilities. So devices are implanted in the spinal cord or in the sciatic nerve to recover the movement of the arms or the legs. They can be implanted in the vagus nerve for the treatment of chronic pain, or there are also retinal implants to recover the vision in patients that are blind. So today I'm going to focus on one specific application, that is the use of deep brain stimulation for the treatment of Parkinson's disease. So Parkinson's disease is a neurodegenerative condition that is produced by the loss of dopaminergic neurons, and these produce motor disorders such as tremor, bradykinesia, rigidity, and postural instability. In the first phase, the treatment of these motor disorders involve the administration of medication such as levodopa, which increase the level of dopamine in the brain. But over time, it is always more difficult to control the doses of dopamine in the brain, and it is very easy to have a peak of overdoses or period where the level of medications are not enough. And in both cases, there are side effects such as dyskinesia, so involuntary movement of the arms. In general, the efficacy of the treatment is reduced over time. In order to overcome these side effects, in the second phase of the disease, the most common treatment is deep brain stimulation, which consists in the implantation of some electrode in a region called subthalamic nucleus. Through a pulse generator, it is injected current, and the current can reduce the motor symptoms, so can suppress the tremor, can reduce the bradykinesia, and at the same time also the level of medications and all the side effects related. So here we have a video of a patient. He has a Medtronic device implanted, and at the moment a doctor has connected the touchpad to set all the stimulation parameters and to optimize the stimulation protocol. And now it is delivered the current. So as you can see, the patient goes from a severe state of tremor to be able to almost completely control the hands. So he can control in a smooth and coordinate way the hands and do also some specific movements such as touching the fingers. So the treatment is very powerful and it works very well. But of course, even in this case, there are some limitations. The first limitation is related to the invasiveness of this lead, which are rigid and also quite thick. So once they are inserted transversally inside the brain, they can produce brain damage. And this opens the need for electrodes that are integrated in flexible substrate to mitigate brain tissue damage and inflammation. The second problem is related to the precision of the stimulation. So usually these electrodes, as the one in the clinically approved leads, they have millimeter scale electrodes and they spread the current in a large volume of the tissue. And this can also produce side effects and compromise the efficacy of the treatment. And also the material are not very high performing material, so sometimes they cannot inject the current that is needed to reach the neuromodulation threshold of the tissue. And finally, the resolution of the recording. So ideally, it would be good to be able to record activity of single neurons and all the biomarkers related to action potential and spikes. But when if a lead has a millimeter electrodes, it can only record the local field potential, so activity of large groups of neurons, and only biomarkers in this specific frequency range. So, in order to overcome all these limitations, in my group, we are exploring the use of a new material, which is nanoporous reduced graphene oxide, on a flexible substrate that is a polymer called polyamide. So this material is produced by filtrating a solution with dispersed graphene oxide flakes to create a thin membrane, which is then hydrothermally reduced to create basal plane defects and oxygen vacancies. This creates a porous structure, which confer to the material special properties that I will describe later. Then the fabrication continue with the transfer of the membrane on a 4-inch wafer with polyamide and gold. And then with photolithography process, we define the electrodes shape and the metal tracks. And at the end, you can see a picture of a finalized wafer with 44 working devices. So then the idea was to explore the potential benefit of reduced graphene oxide for deep brain stimulation in Parkinson's disease. And for this reason, we designed a lead for the rat model, so to conduct some experiments in the rats. And we designed a very compact lead of 120 micrometers in width and only 10 micrometers thick, with an array of 8 electrodes, so 25 micrometers. So this device is a thin film flexible device, is very compact and flexible, and this is very important for minimal invasive neural interfaces. The electrochemical characterization of this electrode shows that for typical stimulation protocol as the one used for deep brain stimulation, so biphasic current pulses of 100 microseconds at increasing current amplitude, we can inject up to 2.3 millicoulomb per centimeter square, so a current density of 21 amps per centimeter square, which is a very high value compared to the standard platinum electrodes that are available in the clinics. So this is due to the special structural and electrical properties of the electrodes, because the porosity of the electrodes increase the active surface area. So even the area of the pores is accessible to the ions. In this way, the electrodes they behave as capacitors, so they can store and inject charge, and this bring to very high charge injection limit. The average impedance spectrum also shows good values, so capacitive behavior at low frequency and low resistive behavior at high frequency. And in subpanel D, we have also some extracted values for 10 different arrays um, with the impedance at 1 Hz and at 1 kHz. So the fact that these electrons can perform in this way allows for the miniaturization of the electrodes. Because even if the electrodes are very small, like 25 micrometers, so the size of single neurons, they can still deliver high charge density to reach the neuromodulation threshold of the tissue, and at the same time are able to record with low noise, so high signal to noise ratio. So the importance of having a flexible substrate is due to the fact that rigid implants over time they form they have some problems, like after a few weeks or 1 month there is the formation of a glial scar around the device, which reduce a lot the quality of the recording and also the capability to inject current, so to deliver the therapy. This is due to the mismatch between the young modulus of this rigid device and the brain. And also other is due to other factors like the size of the device, the insertion force, or the insertion speed. New materials are nowadays used as a flexible and soft substrate for electrodes like hydrogel, PDMS, or Parylene C polyamide S28. But there is also another requirement, that is the fact that this device they had to be inserted inside the brain, so they need to have enough bending stiffness to penetrate the brain tissue. For this reason, just for the insertion of the flexible lead, we temporarily glue the flexible device with a rigid shuttle made by silicone. So one time that the insertion is completed, the glue that is biocompatible and dissolvable, it is by applying PBS, it is dissolved, and the shuttle is removed, leaving inside only the flexible lid. So it was observed that usually during the extraction of the shuttle, the friction between the shuttle and the device was producing a displacement of the device. And to overcome this limitation, we designed an anchoring system made by flaps. And these flaps, they bend only when the device, when it is applied a force to the device to pull it out from the brain. Um, so in this way, the electrodes, they keep staying in place. And this is very important, in particular when it is necessary to target very small and deep brain nuclei. So one time that the insertion is completed, we analyze the recording capability of the electrode. And in this specific case, in subpanel A, we have the recording with one electrode filtered for different frequency band when the rat is alive and post-mortem. So post-mortem, we have a baseline that is just noise, while in vivo you can appreciate from a qualitative point of view that there is much more activity, so there is biological content, that is the real signal that we want to measure. To quantify this difference, we plot also the signal to noise ratio that shows values above 10 for all the frequency range. The combination of very small electrodes, densely distributed, and also capable to record with high signal to noise ratio, allows the localization of the subthalamic nucleus by looking at the recording activity through the different electrodes of the array. So the subthalamic nucleus is very small in the rat, is only 200 micrometers, and it is also placed quite deep in the brain, around 7.7 millimeters. So it is a very difficult target to reach. By looking at but the subthalamic nucleus has also a very particular signature in the activity, and it has a very intense spiking activity, while the region before and after are silent. So by looking at the activity, brain activity in this frequency range, we were able to position two electrodes in the subthalamic nucleus that are the one that you can see in the red trace that are more active, while the others are silent. On the other side, by looking at the local field potential, that is the kind of signal the large electrode can record, it is not possible to extract any information about the position of the lead because the recording is quite similar through all the electrodes of the array. And finally, we have a color-coded spike rate, which quantifies the spike activity in the different channels of the array. And as you can see, the last two, they have much more intense activity that are the two electrodes that are placed in the subthalamic nucleus. Then we try to evaluate the presence of electrophysiological biomarkers related to Parkinson disease by implementing the six-soda rat model. This is done by injecting a neurotoxin in one of the two hemispheres that produce the death of the dopaminergic neurons. In this way, it is possible to simulate the degeneration of the neuron also in the human model. The difference in the content of dopamine between the hemisphere that is injected and the one that is intact, it is possible to be evaluated by looking at the fluorescence, which react with the dopamine. So as you can see, the hemisphere injected is darker compared to the other one, while in a healthy controlled rat, both hemispheres they present the same content of dopamine. This this injection, so the death of the dopaminergic neurons, produce also in the rat some motor disorders as in humans. And in the specific, the rat, they cannot use one of the two paws, the one in the opposite side of the hemisphere injected. There is also, we were able to find a biomarker in the electrophysiological activity of the brain in the subthalamic nucleus, which is not present in the healthy animals. And this biomarker is this very intense burst activity. So the neurons, they fire and they produce action potential, they produce spikes, and these spikes, they fire aggregated in groups that we call burst. So the signal is very synchronized, while in the case of the healthy animal, the spikes are randomly distributed and they follow a pattern distribution. So this difference can be quantified over time by looking at the interspike frequency. So 1 over the interspike time, the time between two consecutive spikes. And in the case of the Parkinsonian animals, there are two peaks. One sharp peak at high frequency associated to the burst, so to the biomarker, and then a hybrid tonic peak, lower frequency, associated to this second component of tonic spikes. While in the case of the healthy animal, there is only one peak at a low frequency. This result was quantified across several animals for both groups. And as you can see, there is a significant difference in the presence of burst between the two groups, while for the tonic spikes, there is no difference. Same analysis was conducted also by looking at the local field potential. And in this case, we were not able to find any significant difference between Parkinsonian and healthy animal. So basically, in the kind of signal that big electrodes can record, it is not possible to distinguish any biomarker or any difference between unhealthy and sick animal. Then the goal was to try to modulate this biomarker by applying deep brain stimulation. So before the treatment, as you can see, we have this trace that is the burst trace that we previously described. And the same thing can be observed by looking the interspike frequency. So there are two bands. And in the specific, there is the one at higher frequency that is the band associated to the bars, associated to the biomarker. After 1 minute of deep brain stimulation, we look again at the brain activity, and what we observe is that the activity is tonic, so there are less bursts, and the band associated to the biomarker is attenuated. Same result can be evaluated also by looking at the quantification of the number of counts of the interspike frequency, and the peak of the burst is less than half after stimulation compared to the pre-treatment. This modulation is also very localized because we quantified the modulation factor of the brain at different distances from the stimulating source. And what we observe is that when we stimulate in place or after 100 or 200 micrometers, we have a modulation factor of almost 1, so perfect modulation of the burst. While when we go farther, 400, 500 micrometers, the modulation factor is close to 0. So this means that the current is focal and the modulation is localized around the stimulating electrode. Then here we compare the effect of the stimulation on a Parkinsonian animal and a healthy control animal. And as you can see, while in a Parkinsonian animal we suppress this interspike frequency associated to the biomarker, in the case of the control animal, there are no major effects produced by the stimulation. And also another interesting point is that after stimulation, the activity in a sick Parkinsonian rat looks similar to the activity of a healthy animal. So deep brain stimulation acts as a neuromodulation tool and normalize the brain activity to a state that is similar to a healthy animal. Different stimulation protocols were tested to evaluate which is the best configuration to modulate the biomarker. And what we observed is that at high frequency, so above 40 Hz, we were able to modulate well the burst, and also at above or starting from 50 micrograms. So with 25 micrograms, we were not able to modulate the biomarker, but starting from 50, yes. This means that it is required to have a charge injection of at least 1 millicoulomb per centimeter square to be able to modulate the biomarker. And this is very, very high value that, for example, platinum electrodes, the one available in the clinics at the moment, cannot reach. And this is another reason why there is the need of high-performing material to deliver with efficacy the treatment. This biomarker, it's not permanently modulated and it slowly recovers. So as you can see, after 100 microseconds, we have again some dots at high frequency. So the burst gradually increase while the tonic spikes decrease. So basically what happens is that there is again a resynchronization of the signal that pass from a random tonic distribution to a synchronized burst pattern. This means that the best treatment would involve a closed loop system where the biomarkers level are constantly monitored, and when the biomarker goes above a certain threshold, it is delivered the current, and then when the activity is normalized, the current is turned off. And this is very important to don't have continuous current and to avoid side effects in the brain and battery consumption. So to summarize, small electrodes with low impedance and densely distributed allow the localization of the subatomic nucleus where it is applied deep brain stimulation. We were able to detect a multi-unit based Parkinsonian biomarker, and by injecting high density charge, we were able to produce long-lasting modulation effect of this biomarker. This bidirectional capability to detect and modulate the biomarker open new avenues for adaptive closed loop system where the current level is adjusted by looking at the biomarker levels. So this work was already published. So if you want to have more info about this study, you can scan the QR code. And thank you for the attention. If you have any questions, I will be happy to answer.
Thank you very much, Nicola, for the nice presentation. Yeah, it was a really nice study that you have explained. Now the floor is open for a question. Just a kind reminder, if you have any question, either please raise your hand and you can open your camera and speak, or please write them on the Q&A box. So maybe, Nicola, I can start with one question, if you don't mind. Like there are many studies that have been shown the use of B.PSS on top of the electrode. How would you distinguish between the use of graphene and B. And like what would be the main differences on this kind of application?
Yeah, so PDOT is also a competitive material with very good performance, so high charge injection and low impedance. I mean, graphene oxide is a good alternative. It's like a low-cost material. It's flexible, is biocompatible, and we proved also that it has a very good stability. So it allows the to have the implant chronically working for months or years. So it is so specifically about the advantages of for this specific application. It's difficult to answer, but we think that graphene oxide can be a good alternative to other high-performing material available at the moment used in the research field.
Okay, great. Yeah, if there's any question, please write them in the Q&A box. So yeah, it was a nice study. Is there any other intention for the use of closed loop control system? Like, have you did any program method or something like this to do it actively, or are you just proposing to use that for future?
Yeah, we didn't try yet. It's just I need an idea for a future study. We think that it would be nice, no, to try to monitor the biomarker level and to apply current only when is needed. At the moment, different types of biomarker for Parkinson are monitored, but not this specific biomarker. So it's like a new solution for closed loop system. And yeah, it would be interesting to try also to monitor some behavioral changes on awake animals. And it's something that we have in plan to do in the future.
Okay, that's great. Yeah, I do think that closed loop would be also important in such application like that. So maybe there is no more question. Yeah. So with that, I would say thank you very much, Nicola, for the nice talk. Oh, there's one just rising. Yeah, you can open your mic if you, I'll lower your mic. Yeah, you can speak. Jin, I think you have raised your hand. Yeah, please, you can speak.
Hello.
Hi.
Thank you for your wonderful presentation. Actually, I'm just wondering the stability of your actual electron during the neuromodulation. So typically, I think based on the fabrication or the technology or the materials itself. So if you apply the current, for the electrical stimulation, right? So actually, the modules itself is not that stable. So I'm wondering how is this in your case?
So you're asking if by injecting high current, if the device lose stability over time?
Yeah, the stability of the electrode during the electrical stimulation.
Yeah. So at the moment we performed some tests of long-term stimulation, and we observed that it's possible to inject up to 2 billion pulses with a stimulation protocol that inject 1 millicoulomb per centimeter square of charge. So the stability is quite good. Of course, there are different components not to evaluate the stability of a device. Also, the encapsulation of the device has to be stable over time. So at the moment, we are focused on this, on the chronic use of the device to evaluate the functionality of the lead over time. But it seems very promising.
So I'm wondering what kind of parameter are you measuring for the stability test? Is impedance change or is the CSIC change or something like that?
So yeah, we perform the complete electrochemical characterization of the electrodes. So we measure impedance, also cyclic voltammetry, to evaluate the presence of possible Faraday reaction, or also we inject current and monitor the voltage. So the idea is to evaluate if there is, for example, metal corrosion that can bring more resistive behavior in the impedance, and also larger voltage polarization during electrical stimulation. And yeah, like for example, water permeation inside the layers of the encapsulation. And yeah, and also we perform Raman on the graphene oxide to evaluate the structural changes of the material. And yeah, we also perform some bending test. Um, so we look from different angles to the stability of the device.
Okay, thank you. I think that's promise very good. Very good compared to other work. Thank you.
Thank you.
Okay. Thank you, Jen, for the questions. Is there any other question? If not, maybe we close it here for today. Thank you very much again, Nicola, for the nice presentation and sharing your research with us. I think there's one more question, maybe last one. How is the stability of reduced graphene oxide during the stimulation in your case?
Yeah, I mean, during stimulation, stability is quite good. We didn't publish yet these results, but there are a couple of papers that are submitted, so they will go out in the next months, and we have good stability. We can inject up to 2 billion pulses without changing the electrochemical performance of the electrode.
Okay, sounds good. So I think with this question, we can end up the seminar for today. Thanks again, Nicola, for the nice presentation and answering all of the questions. And just a kind reminder that we will have the next seminar in the next 3 weeks. So please stay tuned for the announcement for the speaker in the next few days. Thank you again. Thank you, Nicola. Thank you, everyone, for joining.
Thank you for the invite.
Thank you again. Thank you, Nicola. And then we will see you again in 3 weeks. Goodbye and see you until then.
See you.
Bye.